How the TOR pathway and other epigenetic processes create a queen honeybee. The Apis mellifera queen bee is a single fertile female that lays all the eggs of the colony. All other females are sterile. The differences come about as a larva when being fed the royal jelly for more time than other larvae. Royal jelly is a secretion that is used in the nutrition of larvae, as well as adult queens. It is secreted from glands in the hypopharynx of nurse bees, and then fed to all larvae in the colony of all sexes or castes. After 3 days of being fed royal jelly, worker larvae are fed bee bread. The workers construct special queen cells, and the queen larvae in these cells are fed large amounts of royal jelly but are not fed bee bread or honey. The left figure is the outside view of a queen cell, and the right figure is a picture of the inside of a queen cell, where you can see the royal jelly and larva. Bee bread is processed pollen and honey. P-coumaric acid is present in bee bread and honey, but not royal jelly. P-coumaric acid regulates the genes involved in caste determination. Rearing larvae in vitro on a royal jelly diet to which P-coumaric acid has been added produces adults with reduced ovary development. In this figure, A shows ovary development scale of 1 to 5, 1 being undeveloped to fully developed at 5. B shows distribution of ovaries according to level of ovary development in newly enclosed adult bees reared in vitro on diets with and without P-coumaric acid. TOR is a nutrient and energy-sensing kinase that controls organismal growth. A kinase is part of inhibiting the molecule of interest. In queen-destined larvae, the TOR inhibitor rapamycin induces the development of worker characteristics that are blocked by the antagonist FK506. Furthermore, queen growth is associated with elevated activity of the Apis mellifera TOR encoding gene, mTOR, and mTOR gene knockdown blocks queen growth and results in individuals with worker morphology. Gene knockdown is an experimental technique by which the expression of one or more of an organism's genes is reduced. This figure shows the effect of rapamycin and FK506 pharmacology on caste characters in honeybees. In A through G, vehicle means 2% ethanol in insect saline. FK plus R means sequential treatment of FK506 and rapamycin. R means just rapamycin. The W in D shows a worker as comparison. This figure shows that in queen-destined individuals, rapamycin in A shows prolonged development, and in B, a reduced size at adult emergence. In C, ovary size was not affected by pharmacology, but rapamycin in D shows it induced the worker-specific trait corbicula. The arrow in the top picture shows the location on the hind leg. Then, the pictures of a hind leg basotarsus covered with short hairs in vehicle and FK plus R, and the long corbicular hairs in R and W. Apis mellifera workers in queens are not genetically distinct. Rather, these different phenotypes are the result of epigenetically regulated, divergent developmental pathways. Gene expression analysis showed that queens raised from worker larvae differed from queens raised from eggs in the expression of genes involved in the immune system, caste differentiation, body development, and longevity. DNA methylation levels were also higher in 3-day-old queen larvae raised from worker larvae compared with that raised from transplanted eggs, identifying a possible mechanism stabilizing the two developmental paths. The summary and significance of all this is that the mTOR gene is highly relevant with the growth of queen bees. Also, what the queen larvae do not consume is what makes them into a queen. Furthermore, environmental changes, including space and nutrition, in the commercial honeybee practice results in a suboptimal queen phenotype via epigenetic processes, which may potentially contribute to the evolution of queen worker dimorphism.